Next, we'll discuss how changes in the number of chromosomes that a person inherits can affect gene expression. When mistakes are made in separating the homologous chromosomes and sister chromatids during meiosis 1 and 2, respectively, something called non-disjunction occurs. Here we have a normal example of meiosis next to two different examples of non-disjunction. In meiosis 1, homologous chromosomes separate from one another so that the resulting daughter cells only carry a single copy of each. In meiosis 2, the sister chromatids within each duplicated chromosome separate, resulting in 4 daughter cells that are haploid. When non-disjunction occurs in meiosis 1, the spindle fibers pull one or more of the homologous chromosomes in the wrong direction, causing them to travel to the wrong daughter cell. In this diagram, this little chromosome was supposed to travel over to this cell, but instead it went the wrong direction, so now it's in this cell. This results in 2 daughter cells that have an extra copy of that chromosome, and 2 daughter cells that lack that chromosome altogether. When non-disjunction occurs in meiosis 2, the spindle fibers pull one or more of the sister chromatids in the wrong direction, causing it to move into the wrong daughter cell. For instance, on this diagram, this little sister chromatid was supposed to go this way and end up in this cell, but instead it went the wrong direction and it ended up in this cell over here. This type of non-disjunction results in 2 normal haploid cells, 1 cell that has an extra copy of the chromosome, and 1 cell that lacks that chromosome altogether. The cells formed by meiosis are haploid gametes, in other words, egg and sperm cells. If an egg carrying an extra copy of a chromosome is fertilized by a normal sperm cell, then the resulting zygote will have 3 copies of that chromosome. This is known as a trisomy. On the karyotype, you can see that this individual inherited an extra copy of chromosome 18. This condition is known as trisomy 18 or Edwards syndrome and can be life-threatening, especially when the individual is very young. Now, on the other hand, if an egg cell that is lacking a chromosome is fertilized by a normal sperm cell, then the resulting zygote will only have 1 copy of that chromosome instead of the normal 2. We refer to this condition as a monosomy. On this karyotype, you can see that this individual is lacking a copy of chromosome number 9. This is known as monosomy 9. Now, interestingly, animal systems and animal cells can usually deal with trisomies a little more effectively than monosomies. So most true monosomies end up being fatal. Trisomy 21, also known as Down syndrome, is the most famous example of a trisomy. Individuals with trisomy 21 have an extra copy of chromosome number 21. Now, because they have an extra copy of this chromosome, and because the chromosome carries hundreds of different genes for hundreds of different characteristics, Down syndrome can result in a wide range of phenotypic effects. Individuals with Down syndrome often have broad faces and sort of almond-shaped eyes, and sometimes they have developmental difficulties as well, but this can vary widely from individual to individual. On the left, we have a karyotype from someone with Down syndrome, and if you look closely, you'll see that they do indeed have 3 copies of that 21st chromosome. The graph on the right shows the likelihood of having a non-disjunction event occur with age, and what you can see is that the rate of non-disjunction drastically increases over the age of 40. Now, you usually hear about this happening more with women over the age of 40, but it turns out that non-disjunction is actually just as likely in men over 40 as in women over 40. So why do women get the blame for this? Well, think of it this way. Because women only produce one egg cell per monthly cycle, if there's a non-disjunction event there, it's going to show up in that egg. But men produce 150 million sperm cells per ejaculation on average. So it's much more likely for that egg to be the source of a trisomy or monosomy than that sperm cell, since it's in such a big crowd. Many conditions and syndromes occur because of an abnormal number of sex chromosomes. For instance, individuals with monosomy X or XO are lacking an X chromosome. On the other extreme, we have trisomy X. That's where the individual has an extra X chromosome. Now, because the female system is already used to just using one X chromosome at a time and wadding the other one up into a bar body, it seems that this female system can kind of deal with the lack of an extra chromosomes. In the case of monosomy X, the individual just uses that X chromosome all over the body instead of alternating between one and the other X chromosome. In the case of trisomy X, the individual will often have two of these X chromosomes wadded into bar bodies and will use one X chromosome in certain areas of the body, and then it will alternate between the other two in other areas of the body. So I told you that the female body is a mosaic of these genes on the X chromosomes. In the case of trisomy X, it's just a more complicated mosaic. Males can also have an abnormal number of sex chromosomes. For instance, some individuals have an extra Y chromosome, XYY. This is known as Jacobson syndrome or super male condition. Individuals with this syndrome can have heightened aggression and decreased fertility, among other symptoms. It's also possible to have an extra X chromosome. This condition, XXY, is known as Klinefelter's syndrome. Now, because that individual does have a Y chromosome, it does make the body male, but the presence of that extra X chromosome can actually have a feminizing effect on the body, especially during puberty. So individuals with Klinefelter's can have partial breast development, for instance, during puberty. There are also changes that can occur in the structure of chromosomes that can cause lots of different kinds of problems. Deletion mutations, for instance, can cut chunks out of chromosomes and make them shorter. Insertions and translocations can add chunks to chromosome structures and make them longer. Whenever you have chromosomes that are of differing lengths, they're going to have trouble going through processes such as crossing over. So these chromosomes are going to have difficulty with division, and of course that's going to affect the gene expression of the genes on those chromosomes. All right, you guys, you're finished. You made it all the way to the end of the chapter. I hope you found all of this genetic stuff interesting. I certainly did. Be sure to complete your notes and be thorough and bring them to class, and we'll talk about them there.